Hello my dear students. Today we will discuss about that particular organelle which is only found in plant cell. Yes, I am talking about plastids. They are very large and plastids are only found in plant cell as well as in euglenoids. They are easily observed because they are large in size. They bear some specific pigments. Sometimes they impart their color, they are colorless also. So on basis of the pigment present in plastids, they can be classified into three types. Let us see what are their names. First is chloroplast, second is chromoplast, and third is leucoplast. Chloro here word is related to chlorophyll pigment. Chromoplast to various colors, and leucoplast actually they are colorless plastids. So let us see the detail. Chloroplast contain chlorophyll pigment, and we all know chlorophyll pigment is present in leaves. So the chlorophyll pigment, which is responsible for trapping of light energy, it is essential for photosynthesis, and every green part of the plant, it comprises of chlorophyll pigment. So chlorophyll pigment is present in chloroplast. Later on we will see the detail about chloroplast. Next type of plastid is chromoplast. In the chromoplast, fat soluble carotenoid pigments like carotene, xanthophylls, etc. they are present. Mainly they give color to that part of the plant which are beautifully colored. So they are responsible for giving yellow, orange, red or different colors. Mainly the flower petals, they are colorful. Sometimes the outer skin of fruit or seeds, they are colorful. So all are due to presence of chromoplast. Now last type of plastids, they are known as leucoplast. Leucoplast, as I have said to you, they are colorless plastids. They are of various shapes, size, and mainly their function is to store nutrients. And nutrients in the form of three types they are stored. First is amyloplast. They are responsible for storage of carbohydrates, for example in potato, rice, etc. Secondly, they are elioplast, which are responsible for storing oil and fat like in groundnut seeds or every type of oily seeds. Thirdly, they are aleuroplast. Mainly they are responsible for storing proteins. So protein containing seeds or protein containing parts, they store aleuroplast. So in this way, three type of plastids they are found in plants. Now let us discuss about the description regarding structure and function of chloroplast. Here you are able to see the picture of chloroplast, the 3D picture of chloroplast on your screen. Majorly the chloroplasts are found in green plants or green part of the plant, and that is mesophyll of leaves, mesophyll cells of leaves. So particularly the location of chloroplast is mesophyll. If we talk about the structure, they are lens shaped, they may be oval, they may be spherical, they may be discoid or even ribbon like, means flat. They have variable lengths like 5 to 10 millimeter, width may vary from 2 to 4 millimeter. Even their number, it varies from cell to cell. Sometimes they are present only one in number, like in Chlamydomonas, it is one type of algae, green algae, and sometimes 20 to 40 in number per cell, like in most advanced type of angiospermic plants. And in their leaves, in the mesophyll cells, they vary in number, they are more in number, like from 20 to 40. Now if you see the picture, there are certain labelings which are shown to you. The chloroplast also contains two membranes, outer membrane and inner membrane, just like mitochondria structure. Here inner membrane, they are forming the granular structure which are forming intergrana. Then the space which is present between the inner membrane of the chloroplast, that is known as stroma. If we compare mitochondria and chloroplast structure, the mitochondrial matrix, the cytoplasm of the mitochondria, we called it mitochondrial matrix. Here we are naming it stroma. Then a number of organized flat membranous sac-like structures are present which are known as thylakoids. This term is very important to remember, and they are present inside the stroma. You can see in the diagram. Now if we describe the thylakoids, thylakoids are arranged in stack pile of the coins as we arrange coins one above the other. Similarly, the thylakoids are also present one above the other, forming stack pile. And there are so many grana. Then the membrane of thylakoids encloses a space which is known as lumen. The stroma of chloroplast, it contains many enzymes which are responsible for photosynthesis. Even the chloroplast, it contains its own DNA, that is circular DNA molecule, as well as ribosomes. We have said earlier that chlorophyll pigment is present in these chloroplasts. So where it is present? It is present in thylakoids. The ribosomes which are found in chloroplast are smaller. They are of 70S type. Many times we are using the word ribosomes. Even these ribosomes were present in bacterial cell, means prokaryotic cell. So what are these ribosomes made up of? Actually, ribosomes are granular structures just like granules, which are observed for the first time in electron microscope by George Pallet. They are composed of RNA, that is ribonucleic acid, plus protein, and they are not at all surrounded by any membrane. The ribosomes are found differently in prokaryotic and eukaryotic cells. In prokaryotic cells, they are of 70S type, and in eukaryotic cells they are found as 80S type. So actually they are formed of two subunits, smaller unit and larger unit. Here S stands for sedimentation coefficient. Now let us discuss about cilia and flagella. Actually they are the part of cytoskeleton, and here cytoskeleton is responsible for certain movements in cell. Even you are familiar with about these terms, cilia and flagella. Cilia is a plural word, singular word is cilium. Similarly, flagella, it is a plural word and singular word is flagellum. They are hair-like structures. In bacterial cell, we have understood the importance of flagella. Flagella are actually long structures, thread-like structures, and they are only few in number as compared to cilia. Cilia are comparatively smaller structure, hair-like structures, and they are many in number. Both are responsible for movements or any certain type of activities. So according to book, let us see. Cilia are small structure which work like oars, causing the movement either of the cell or surrounding fluid. Similarly, flagella, they are comparatively longer and responsible for certain cell movements. We have seen the flagella structure in bacterial cell. Now let us see the detailed structure regarding cilia and flagella. If we see the electron microscope study, their structure, it is very simple one, but not easy to understand. It is more understood with the help of diagram. So first of all, we are just observing the important lines regarding the structure. Their core, that is the central part, it is known as exoneme, and it possesses number of microtubules which are running parallel to the long axis. These microtubules, they are very much similar, just like pipes. The exoneme usually has 9 pair of doublets in which are radially arranged and a pair of centrally located microtubule. So such type of arrangement, we call it 9+2 arrangement. 9 outside and 2 in center. So central tubules, they are connected by tubules by radial spokes. So these are radial spokes. So all these structures, they are more clear in diagram. So students, you have seen how the cilia and flagella, they look like, their cross section it looks like. Now we will talk about centrosomes. They are also known as centrioles, and these are only and only organelle which is found in animal cell. So what is the structure of centrosome? Let us see. The centrosome is an organelle usually containing two cylindrical structures called centrioles, and they are surrounded by material. Both the centrioles in the centrosomes, they lie perpendicular to each other, in which it has organization like cartwheel. So this line is very important to learn, and I'll show you how it look like, how they are present just right angles to each other or perpendicular to each other. The centriole structure, it is made up of 9 evenly spaced peripheral fibrils which are made up of a special protein known as tubulin. Each of the fibril, it is a triplet. That means 3 pipe-like structures they are present, and they are adjacent triplets, they are also linked. Even the central part of the centriole is also having proteinaceous hub which is connected with the peripheral triplets by radial spoke made up of protein. That's why the diagram, it looks like the cartwheel. So in this way we have seen the structures regarding cilia, flagella and centrioles. I will show you more clear diagram, and these diagrams which I have just explained you, they are important to draw in the classwork. Okay students, right now I am showing you the structure of chloroplast. This will be easy for you to draw. Here it is showing outer membrane, inner membrane, then thylakoids which are arranged. You can see here I am indicating them with blue color. So they are arranged one above the other just like stack pile of coins. Then even the whole bunch of this thylakoid is known as granum. There are so many grana I am showing here one by one. So all these are grana. These grana, they contain chlorophyll pigment. Now this yellow colored area, that is known as stroma, and here also the essential enzymes which are responsible for photosynthesis, they are present. All this lamellar structure, it is found inside the stroma. So this diagram will be easier for you to remember and draw. Even certain ribosomes are present, its own chloroplast DNA is present, and the basic structure is very much similar to mitochondria. Even the starch grains are present which are helpful in storing the energy, food. Now this diagram, it is showing the 9+0 arrangement which is found in the centrioles. These centrioles, they are found in animal cell only. These A, B, C are 3 subunits which I have just mentioned. It is known as triplet. So this triplet, it is made up of microtubule and which is made up of tubulin. Now these are spokes which I am marking 1, 2, 3, 4, 5, 6, 7, 8 and lastly 9. So these are 9 spoke like arrangement. These spokes are also present in the center, but these 9 are triplets which are arranged. In centermost part, which I am shading with yellow color, that is showing the proteinaceous hub. All these tubules, they are connected with the center also and in adjacent areas also. And here this diagram is very much similar to the structure of cartwheel. So this is its identification. Okay, and even you have to remember 9+0 arrangement, 9 in the outer peripheral region and here we are able 0 in the center. 9 doublets, these are peripheral fibrils, microfibrils. This diagram is not important to draw, but you have to only understand. A and B microfibrils. So here it is known as doublet. Even they are linked with one another here, 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 all of them, they are linked here. Even in the centermost part here, these central fibers, they are located. The two myofibrils, they are present in the center also, and this particular central and peripheral myofibrils, microfibrils, they are connected with the spokes. So these are spokes. So here the arrangement of microfibrils, they are known as 9+2 arrangement. So this is the original picture which is showing the 9+2 arrangement of cilia. So I hope you enjoyed today all these organelles, their detail. Please revise all of them, draw the diagrams in your classwork, do revise it daily, and today we end the session here. Till then, goodbye students.